Good morning, everyone. I shall be presenting my Afmrc project titled Therapeutic Drug Monitoring of Imatinib in Patients of Chronic Myeloid Leukemia Chronic Phase and its correlation with molecular response. Imatinib is the gold standard for treatment of chronic myeloid leukemia chronic phase. Pharmacokinetic studies have shown variability in trough concentrations of imatinib due to variations in metabolism, poor compliance, or drug-drug interactions and may impact clinical response. Therapeutic drug monitoring provides added information on efficacy, safety, and compliance than clinical assessment alone and help in clinical decision making. Trough imatinib plasma level of more than equal to 1000 nanogram per ml correlate with better cytogenetic and molecular responses. It is relevant for patients with suboptimal response to treatment or treatment failure, severe adverse events, possible drug interactions, or suspected non-adherence. Further prospective studies were needed to confirm the relationship between imatinib plasma levels concentrations with response and to define effective trough imatinib plasma levels. The aim of my study was to study the plasma trough levels of imatinib in CML-CP patients. The objectives were firstly to correlate imatinib plasma level with molecular response on at least two occasions, to study the variation in trough imatinib plasma levels during treatment of CML-CP with a standard dose imatinib of 400 mg once daily. Thirdly, to establish the therapeutic efficacy of cheaper generic imatinibs. Fourthly, to establish the protocol for therapeutic drug monitoring of imatinib by liquid chromatography mass spectrometry protocol and achieving the recommended drug level to maintain a sustained major molecular response. The study was approved by Institutional Ethical Committee. 50 chronic myeloid leukemia patients were enrolled from 1st July 2017 to 30th September 2019 with written informed consent. Optimal drug compliance was ensured before and during the study. Patients received generic imatinib available at different times during the treatment. Those included were CML-CP patients diagnosed at least 12 months prior who were treated with standard dose imatinib of 400 mg once daily and had achieved a major molecular response by molecular monitoring of BCR-ABL transcripts in international scale with real-time PCR. Those excluded were patients who were on higher dose imatinib of more than 400 mg per day or patients in accelerated phase or blast crisis, any history of blood component transfusion within the past 3 months, any history of coexistent malignancy, chronic liver disease or chronic kidney disease, those with poor compliance or non-adherence, that means missing of more than 3 doses of imatinib per month, were also excluded, and those detected with BCR-ABL kinase domain mutations if they had not achieved their target cytogenetic or molecular response time points during the treatment of CML-CP with imatinib. In the methodology, 3 ml blood was collected for monitoring of trough imatinib plasma levels in plain sterile vacutainer before morning daily dose at steady state. Blood samples were collected twice at interval of 12 months, at least more than 3 months apart. On the CML day, a medical camp was conducted to do so on 22nd September 2018 and 22nd September 2019. 3 ml blood in EDTA vacutainer was simultaneously collected for measurement of BCR-ABL transcripts by real-time PCR. The total RNA was extracted from peripheral blood cells and BCR-ABL transcript levels were quantified using real-time PCR. BCR-ABL percentages on international scale were interpreted as less than 0.1% or 3 log reduction suggestive of a major molecular response, which is a therapeutic target in CML after 12 months of imatinib therapy. Less than 0.0032% or a 4.5 log reduction suggestive of a deep or complete molecular response. The trough imatinib plasma levels were determined using liquid chromatography mass spectrometry protocols, which included high-performance liquid chromatography coupled with tandem mass spectrometry and has been previously validated in countries outside India. The trough imatinib plasma level measurement has been performed only at 3 centers in India prior to our study, and they needed further validation and stringent standardization. Scheme of steps involved in the extraction of drugs from serum is as shown. 200 microliter of serum was mixed with 800 microliter of chilled methanol. Then the sample was vortexed, mixed and centrifuged, and the supernatant was added to the Captiva ND lipid cartridges and further injected into the LCMS system. This is the protocol validation which was done at Biochemistry Laboratory at Institute of Aerospace Medicine, Bangalore. The multiple reaction monitoring conditions in LCMS system and their values are as shown in the slide. The further calibration curve for imatinib was plotted, and for each patient the plasma levels of imatinib was plotted on this calibration curve. A total of 50 CML CP patients were enrolled and 30 were included. 20 excluded were the 7 declined to participate in the study, 5 had imatinib failure during the course of their treatment, 4 required escalation of imatinib dose, 2 patients progressed to CML accelerated phase, 1 patient did not tolerate imatinib, and 1 patient proceeded on to treatment-free remission. These are the baseline characteristics of the study patients on standard dose imatinib in major molecular response. 19 were males and 11 were females. 53.2 was the mean age with a standard deviation of 13.9 of the study patients. The molecular response at 2 time points of MR1, MR2 and the average MR molecular response is as recorded in the table. Subsequently, trough imatinib plasma levels are as mentioned in the table. All our patients achieved a level of more than 1000 nanogram per ml. Same imatinib levels and BCR-ABL levels at 2 time points and average levels have been plotted into box and whisker plots. Further, a comparison of two groups between the major molecular response and complete molecular response was done, of which 19 achieved a major molecular response and 11 achieved a deeper complete molecular response. The age, the trough imatinib plasma levels and molecular responses are as mentioned in the table. The receiver operating characteristic curve for imatinib levels on 2 occasions as a determinant for major molecular response are as plotted. Therapeutic drug monitoring is an important tool in the management of chronic myeloid leukemia, and there is a close relationship between drug levels and clinical outcome. Imatinib plasma levels are useful to evaluate patient compliance, potential drug interactions, treatment efficacy, and severe adverse events. Therapeutic drug monitoring is useful in dosage adjustment of drugs with marked inter-individual variability or narrow therapeutic window. Rough imatinib plasma levels may help as a marker for suboptimal response and may identify patients in whom increase of dose or change in therapy is indicated. Average mean trough imatinib plasma levels more than 1000 were achieved for all patients. Our results were in concordance with the previous studies. No publications from India regarding compliance to imatinib have been documented, but data from the West indicate it to be a major issue in suboptimal responders. Individual patient characteristics including age, gender, race, weight, and inherited differences in drug absorption, metabolism, transport, disposition of drugs, and interactions between prescribed medications may also affect drug levels. These are the different studies with which our study has been compared for imatinib levels. To conclude, trough imatinib plasma levels in therapeutic range influence molecular response in chronic myeloid leukemia chronic phase patients and is an independent predictor for sustaining depth of response. Adequate plasma concentrations of imatinib are essential for optimal clinical response. Generic imatinibs are bioequivalent and comparable in clinical efficacy, help in saving cost, and are efficacious for sustained major molecular response in CML-CP patients. Monitoring of imatinib plasma levels may be used for all CML patients or at least for treatment failure or suboptimal response. The study was supported by an Armed Forces Medical Research Committee and has already been published. My co-worker Wing Commander Ashwini Kumar helped in conducting the lab part of the study. The Colonel Anurod Gupta helped in outlining the initial methodology, and last but not the least, the idea and conceptualization of the study was done with the help of Group Captain TVS VGK Tilak. These are the references. Thank you.